Hello, today we are going to discuss one of the experiment, that is the introduction to hemocytometer. It is one of the practical is mentioned in your syllabus as per the Savitribai Phule Pune University or as per the PCI syllabus. So introduction to hemocytometer. It is one of the kit which contain, here I will show you. So this is one of the important chamber, okay, that is called as a Newborn's counting chamber. So this Newborn's counting chamber, here you find Newborn's counting chamber. Okay. So this Newborn's counting chamber, it is used to determine red blood cell count, white blood cell count and platelet count. Then it also contains two pipettes. This is the one of the pipette. Okay. Here you will find these are WBC pipette, white blood cell pipette. Okay, which is that bulb. Here in the center of this bulb there is a bead is available which is a white in color. Okay, and if you see here, so the pipette, so up to 0 to 11, 0 to 11, that is a marking is available. Okay, and here is a white bead is available in this bulb. Then another pipette is a red blood cell pipette, which is that tip is a red in color. It also contains the bead which is a red in color. If you see the marking 0 to 101. Okay, so you have to suck the blood up to 0.5 in the both the pipette. For determination of white blood cell, for determination of red blood cell, you have to suck the blood up to the 0.5 mark. Okay, it is a very simple experiment. So hemocytometer, so it contains Newborn's counting chamber, Thomas cover slip, which is a cover slip which is a place on this Newborn's counting chamber. See here is a Newborn's counting chamber. There is some ridges are there. Okay, there are these two blocks are available here, this one and this one. At the center there is lines are there. Okay, which is it could not be visible. Okay, so under microscope you have to observe this. Okay, and it contains two pipettes. One is RBC pipette, WBC pipette, again Newborn's counting chamber and Thomas cover slip. So these are the apparatus which is available in the hemocytometer kit. Okay, thank you. And in case of RBC 101.5 to 101. Okay. Adjustment of Newborn's counting chamber. First of all, you have to clean the microscope. First of all, means what are the useful hints that I will explain you. So before counting the Newborn's, before adjusting the Newborn's counting chamber, you have to always clean and adjust the counting chamber, cover slip, microscope and the pipettes before pricking the finger for blood collection. Okay, suck the blood and dilution fluid slowly, uniformly and exactly to the mark. After sucking the blood up the required mark, remove the extra amount of blood from the tip of the pipette by cotton, gently and carefully. A knot may be given to the sucker tube after sucking the dilution fluid up to the particular mark. If you are not placing the knot, it's well, but try to bend the sucker of that particular tube. The care should be taken that the fluid does not come out from the either end. One end may be closed by putting the finger at the tip of the stem while putting the knot. Never forget to discard the few drops of fluid in the stem of the pipette because it does not contain the blood. The fluid should not enter into the grooves of slide. Okay, and if it happens due to the capillary action, it will be draw cells and thus giving the error in the counting. So it is advisable to form a drop at the tip of the pipette and the drop should simply touch the slide. The fluid will spread automatically due to the capillary action. The cell must be allowed to settle for a period of 2 to 3 minutes before starting the actual count. For counting the number of cells, prepare a table consisting of the column and enter the number or cell in each column. The cell touching the two boundaries are to be counted, while cells touching the two boundaries not to be counted. So these are the some useful hints. So these are in chamber you have to clean the microscope. Okay, now you have to first of all, before adjusting the microscope, before adjusting the Newborn's counting chamber, you have to clean the microscope properly. You have to, with the help of this 10x, these are 10x lens, these are 40x lens, okay, and this one is the 100x. You see here, these are oil immersion lens, okay, these are 40x, okay, and here I have, first of all, before you see the distance between the glass slide and objective, okay, so this is very important. Don't touch the objective to the Newborn's counting chamber, okay. So before adjusting the Newborn's counting chamber, means before counting the cell for WBC and RBC, so one of the difference is there. For WBC you have to keep the objective at 10x, for RBC you have to keep the objective at 40x, okay. But before, for adjustment of the RBC or for adjustment of WBC, first you take the help of 10x objective lens, then in case of RBC you have to switch over to 40x. Now you see the difference. Yesterday I have told you, so in the previous practical I have told you about the experimental part, okay. Now there is a distance between the objective lens and the Newborn's counting chamber, okay. Okay, so here I will show you under the microscope with the help of mobile camera measurement, RBC measurement, okay. Now we are observing at 40x, okay. These are, now we are observing at under 40 when we are observing at under 40x, then it will be a look bigger one, but now we are observing under the 10x, so it will be a very smaller in size, okay. These squares you have to observe under the microscope at 10x power objective, okay. These are left upper. Now I will switch to the right upper.